if you're the only.
We have Dr. Dan Selbie from the Department of Fisheries and Oceans, Quesnel Lake Sockeye. This is a little lab that Dan has taken over that has a very long history of research in salmon biology and so on. And I found out about Dan from the Pacific Salmon Commission staff, who he gave a seminar to last fall, and they said it was one of the best seminars they'd ever heard, so we contacted Dan and asked him to come and talk to us. Dan did his undergrad at Queen's University at the same time that my son Dan was across on the other side of the bay.
RFC.
Yeah. And his PhD at Queen's as well, a postdoc at McGill, and then came west. And he has adjunct appointments at SFU and the Fraser Valley University. So he has academic as well as government connections. And he's going to talk to us about one of the biggest puzzles in fish biology, what's going on in Quesnel Lake, one of our biggest sockeye stocks. Dan,
thanks, Carl. Right on. Really appreciate the opportunity to come here today. I do, especially since we started down this road, do get the opportunity to reach out to a number of different groups, in this case university, which is great and such a venerable institution. I want to preface the talk by saying this is by no means a complete story. As Carl says, people have been wrestling with what's happening with large escapements in the Fraser and in particular Quesnel Lake for some time, but I think we're starting to look at data that we have in new ways, and that might make us think about these problems a little differently than we have in the past. So again, talking about Quesnel Lake Sockeye Salmon, which used to be a big producer in the Fraser, now somewhat infamous for other events that have been going on in the watershed, but large escapements, some trophic surprises, and the consequences of density dependence. Just as far as the talk overview goes, I want to start with talking about the recent context of escapement of sockeye in the Fraser. I'm not sure what people's individual backgrounds are here. We've seen some large escapements in recent times, and one might say that we're in a new era of production in the Fraser, at least within the recorded window. A little primer on nursery lake ecology. Of course, sockeye salmon amongst the Pacific salmon species are the example of fish that use nursery lake ecosystems to rear their juveniles. And talk about top-down versus bottom-up processes in those environments. Marine-derived nutrient fluxes, which probably most people are familiar with now that research was really in vogue in the early 2000s. Focus in on our work from Quesnel and some of the evidence we have on phenology. So this is lake ecosystem structure and functioning, juvenile salmon ecology within Quesnel Lake, and the potential for population feedbacks associated within lake processes. And then I just want to touch, this talk is a sort of iteration of one I've been giving to management people in DFO and beyond, talking about what happens when you get up to large escapements in species such as sockeye salmon. Focus on the Fraser system in the context of areas where there are lots of fish, and that would be Alaska. So that's the general overline for the talk today. I'll launch into it. You know, sockeye salmon in general are quite diverse species. They're spread throughout the Pacific Rim. In Canada, the majority of the conservation units for sockeye use a lake-type existence, as I mentioned. Some use main stem river environments. But you can tell by this plot that there's an incredible diversity biogeoclimatically, as well as hydrologically, where these fish are existing. And in Canada, we have over around 216 lake-type sockeye. So it keeps me busy. We're pretty much the only group that works on lakes within DFO's science branch, and certainly on sockeye lakes. Most people here are probably familiar with the fact that these fish are anadromous, meaning they reside in freshwater and marine environments to complete their life history. Juveniles, again, for some period, are born in freshwater and rear in there for any number of years. For sockeye, it's anywhere from 1 to 3 years, or in the case of Harrison sockeye, they move right into the ocean, bit of an anomaly. Accrue 95% of their biomass in the marine environment, so they're actually picking up a lot of nutrients that are sequestered in their tissues. And then these nutrients, again, because they're semiparous, are delivered to freshwater ecosystems. That's this marine-derived nutrient flux or nutrient cycle, or nutrient pump, it's often called, associated with salmon. So the picture in the Fraser is variable. This is a system where we see cyclic dominance in a number of stocks. That's often in the Fraser 4-year cycle where you have a dominant line, and you may have a subdominant line the year after, followed by fallow years of lower production in these individual rearing environments and stock productivity, associated stock productivity. You can see here, the only difference between the green and the black is this is based on reconstructed values going back to the late 1800s. This figures from the Cohen Inquiry main report. And I would argue that, at least in a scientific context and in a fisheries management context, we're entering a new era in terms of overall large escapements and production. You can see that there's been large builds on dominant cycles throughout the 90s, and then some extreme variability, of course. Recently, I don't have past, I think, 2011 or something, or 2010 there on this plot. But nonetheless, we've had some spectacular variation, as you probably well know, precipitating judicial inquiries and also boom and bust in terms of fisheries on Fraser salmon. This issue of large-scale population variability, or large escapements especially, influencing having negative feedbacks on stock productivity has been a debate that's been going on for a long time. We know density dependence exists. The question is, do other processes like delayed density dependence, cycle line interactions, result in stock collapse? And one of your folks in the room here today, Carl Walters, led this particular submission looking a number of years ago at whether or not these large escapements can cause stock collapse. Of course, this issue hasn't gone away. There's been declines identified, of course, here in the Cohen Inquiry, declines in productivity through time. So this is just recruits per spawner across a gradient of time, brood year plus 4. You can see there's been these recent declines. And so there could be a number of reasons underlying these recent trends, but the issue of large escapements has not gone away. In fact, it was a focal point of one of the main technical reports of the Cohen Inquiry, and this is work that was led by Randall Peterman and Bridget Dorner. And so to bring in the Quesnel context, this is the one stock where the statistical modeling based on stock recruit showed cycle line interactions. So interactions between the dominant and subdominant lines and those fallow year lines, those non-dominant lines. So real strong evidence for this, fit with Larkin models, if you're familiar with models that explicitly incorporate cycle line interactions. And so Quesnel was the one that stood out in the, I think, 21 CUs within the Fraser. I think they looked at 19 that showed strong cycle line interaction, especially at large escapement. So just as far as the factors and processes on the freshwater side of life history for salmon that influence stock productivity, they can be split into two groups, density dependent and density independent, although some of the factors here could be considered density dependent as well. Density dependent just means the number of fish influences the mechanism of influence. And so in terms of limitation, in lakes and rivers, you can have spawning limitation. There's only so much available habitat for fish to spawn, and therefore if you have access to that, you may start to erode productivity by ripping up redds and redepositing eggs, etc. Rearing limitation. So again, especially for sockeye, nursery lakes tend to be the focus of that work. Are there factors trophically within nursery lakes which limit salmon production? And then all these other suite of factors, so disease, interspecific competition, say with kokanee or other pelagic planktivores in lakes, predation, which is again not necessarily density independent, environmental variability and environmental stressors. All these factors, and probably a whole suite more, have strong influences on survival and growth of fish. And so it's no small wonder that when you, say, put together a judicial inquiry to find the smoking gun, there's a potential for going down a lot of paths, and you don't necessarily find the answer you're looking for. So the other thing I want to highlight here, and I'm talking mainly from a freshwater perspective today, but is the potential for interstage effects. Effects that can happen in freshwater that carry over to the marine environment and influence your survival once you go out to sea. Because it can be either a hospitable or inhospitable environment depending on a number of factors in the Pacific Ocean. today I'm going to be focusing principally on rearing limitation issues in freshwater lakes, because that's what we do. So this slide I actually put together a number of years ago now, leading into the bilateral review. Scientists from the US and Canada were called together to look at the issue of the low returns in 2009 in the Fraser. And so it turned out our group had produced a model. It's a very simple photosynthetic rate model, which predicts rearing capacity in terms of juvenile densities in nursery lakes, and also optimal spawner returns. And so what you can see here is the green bars are the maximum observed spawners, again up until 2010, observed in the Fraser for major systems we've studied, and the relative habitat productive capacity models beside it. The take-home point here is, relative to habitat capacity model estimates, and in many cases stock recruit estimates, we do see exceedances of these optimal capacities in these environments. And so it sets the stage for potential for feedbacks to stock productivity. Again, I'm not sure of the diversity of backgrounds in the room, so I put in a little primer on lake ecology, talking about the principal top-down and bottom-up stimulus or forcings within these ecosystems. And so, in a general context, you can put bottom-up forcings into nutrient stimulation. This can come from a variety of sources. It doesn't have to be marine-derived nutrients, but often it is. And that is oppositionally forced by some top-down processes. In this case, I've chosen sockeye planktivory, so feeding heavily cropping down zooplankton populations and perhaps interrupting vertical flows of nutrients. There seems to be vertical flows of nutrients or energy flows to higher trophic levels. No, it's okay. I did it, it seems to be. Well, maybe not. I don't know. Carry on. So, I mentioned this research on marine-derived nutrients has been a long-term process. It really became in vogue with the work by David Suzuki, the salmon forest, bringing it into the limelight. And in my earlier part of my grad career, actually, there was a huge conference convened in Eugene, Oregon, looking at all these things. And lo and behold, there's a wealth of evidence to suggest that there's a stimulatory effect of nutrients on freshwater ecosystems. Some seminal work by the Russians back in the 70s looking at nutrient loading to sockeye lakes. I think these are Lake Dolny and Blizny in Russia. This is in Kamchatka. You can see as salmon escapement increases along the x-axis, you have an increase in phosphorus loading to some asymptote. But nonetheless, an enrichment that seems to be related to phosphorus, or related to salmon carcasses and phosphorus. And also, there's a commensurate figure in that report, I think, that shows chlorophyll response. So an algal primary production response to these increasing salmon carcasses. That was kind of early days. This work formed the basis of a lot of the experimental lakes fertilization, or experimental fertilization of sockeye nursery lakes, both in Canada and the US, the FRED program in Alaska, and here the Lakes Enrichment Program. And it was generally concluded that there was increases in smolt biomass associated with increases in nutrient loading. Generally considered to be a positive effect, although there was a great degree of success in terms of accruing smolt biomass associated with those enrichment additions, probably related to local conditions, the variability of local conditions in which they were applied. So coastal lakes tend to be very nutrient poor and highly flushed, versus interior systems that retain nutrients and have longer residence times. So that's the top-down bottom-up story. Now focusing on top-down, we do have pretty good evidence that there is top-down forcing in sockeye nursery lakes. These are just growth relationships across a gradient of effective female spawners, looking at either circuli and otoliths or fall fry weights as measured using hydroacoustic control surveys in freshwater lakes, nursery lakes. And for a number of the big systems, I showed Quesnel, Shishwap, Quesnel, and Chilco here. These are historically the big producers on the Fraser. A negative effect as you put more fish in these lakes on growth. That could, we've looked at this in terms of survival. These are just juvenile fall fry abundance or smolt abundance at Chilco relative to the number of effective female spawners put into these systems. So number of juveniles levels off or even starts to decline as a result of the number of fish that are in the system. So this is inferred to be a density dependent or a top-down effect on survival. The story, of course, is both. We published a paper at Oikos a number of years ago where we were looking at long-term paleoenvironmental archives and reconstructing salmon abundance and zooplankton abundance in these systems. And this is for Alaskan systems, but you can see that over the lower density of inferred spawner abundance, there may be a positive effect, but really when you get out to those high density systems, and I think this is Red Lake, Alaska, which receives somewhere like 40 or 50,000 spawners per square kilometer, really high density sockeye nursery system, you tend to erode those bottom up processes and resources available for salmon, in this case, zooplankton. So I promised to talk on Quesnel Lake. That's just kind of a backdrop to some of the things I'm going to be talking about. Quesnel Lake, as I mentioned, a long-term important producer within the Fraser. Not so much in the last decade. I'm hoping to talk about reasons behind that. Interestingly enough, in the last year, Arl and I were just talking out in the hall that the preliminary estimate is almost 1 million sockeye moving into Quesnel Lake, and that may change as they finalize the stock assessment information. But nonetheless, this system seemed to be broken for quite a while, or at least the stock seemed to be broken. Really depressed abundances for quite some time, and this may be good news starting this year. Sadly, coincident with some other extraneous events, specifically referring to Mount Poly mine disaster, but that may have influences on that, or may not. We'll see. So it's a large fjordal system. Fjordal just means narrowly constrained by mountains, and it's a multi-arm system. So I'll refer to spatial and temporal variability in some of the measures we've taken from Quesnel Lake here. There's the east arm, which tends to be quite an oligotrophic or nutrient-poor rearing environment. The north arm, which has been increasing in nutrient status, and the Mitchell River stock, which is a large component of the Quesnel complex, pours into this area of the lake. And then the main arm, and really some people call this the west arm, receives both nutrients and, of course, juveniles that are in the Horsefly River, which tends to be a large producer as well. The lake also has shore spawning populations throughout the eastern margins. So, but it's important to realize that the fish don't necessarily stay in one arm, and we do have evidence, that they may go where the groceries are. Again, a somewhat incomplete stock assessment history here, but nonetheless, showing those massive returns in 2001, 2002, unprecedented in recorded history, followed by great declines in stock abundance, which carried through again, as I mentioned, until roughly this year. There we go. This is also represented as a nutrient pump or nutrient loading, aerial nutrient loading as well to the lake, and so just based on average carcass composition of nitrogen and phosphorus, we show substantial changes in nutrient loading to Quesnel Lake. So you would, going back to your trophic ecology, say, oh, this has got to be good for sockeye. I just want to use this slide to orient you on a number of bar graphs I'm going to be presenting going forward. Some people tend to get lost in the details, and I hope to avoid that here today. On the x-axis of a number of these plots, it's broken down spatially by the east arm. Of course, I got it backwards here. East arm, the more oligotrophic arm, the main arm, and the north arm. So we have a number of limnological stations that were shown on that previous map that represent spatial variability in nutrients, plankton within lakes. So our program looks at physical, chemical, and biological variables within these lake ecosystems as they support sockeye, and then of course juvenile abundances using hydroacoustic control surveys. On the x-axis would be some variable, but I just want to say that within each one of these clusters by space is a before period, a lake period experienced by the peak escapements. In this case, we had full limnological workup in 2003, which is the year where the subdominant line, the major subdominant line was rearing within the lake. So you can consider that an impact years. The black bars are impact, and on either side before and after. All right? So hopefully that's clear. Okay, so this is a panel that's clearly set up for a publication and not a talk, but nonetheless, I just want to walk through some of the responses that we see in nutrients and primary productivity within the system. So again, across that east main and north arm gradient, you see that there was a stimulatory effect on phosphorus in each of these major basins. And we believe that was generally associated with fish, but I'll hope to convince you later. And it seemed to be aberrant compared to periods prior or after the major escapement in 2003 lake here. Looking at other sources of nutrients that are important to aquatic ecosystems, if you look at seasonal nitrogen or nitrate minima, plants, just like your grass on your lawn, needs nitrogen and phosphorus, phosphorus typically being the limiting nutrient, but nitrogen gets soaked up when lakes stratify. So, there's disproportionate warming of those surface layers of the lake, and you actually get recalcitrance to mixing between surface waters and deep hypoluminetic waters in the lake. And you can see within the area where phytoplankton are operating, a strong depletion of nitrate. So that suggests that there was a strong photosynthetic response associated with nitrogen loading. Looking at silica, which is important to a particular group of algae, the diatoms. They make and I'll talk a little bit more about that later, but they make their cell walls out of glass. They take soluble reactive silica from the water and create very ornate glass cell walls. You can see that there was major drawdowns during that peak escapement event as well, showing a strong primary productivity response anyhow. If you don't believe me from the nutrient picture, you look at either surrogate of standing algal biomass in chlorophyll A here, or in photosynthetic rates. We're one of the few groups that actually does C14 uptake experiments to get at rate information. You can see that there was a substantial change in the amount of primary production, primary productivity that was going on in the Quesnel system following those peak escapements. In all cases, you saw a decline afterwards in either nutrient status or in the case of phosphorus or primary production. Quesnel Lake is a very, very deep lake, and so you likely have a lot of loss of nutrients that are delivered to surface waters to those deep environments. It's just simple volumetric dilution. So we can use tracers like stable nitrogen isotopes to track the fate of marine-derived nutrients in these systems, and we looked at enrichment in plankton communities in the zooplankton, and also at the higher trophic level in sockeye. And you can see that the underlying theory here is that sockeye are enriched in N15 relative to freshwater environments, and so you actually have an enrichment when marine-derived nutrients come in. This seemed to translate into incorporation at both the zooplankton and the juvenile fry levels. So these nutrients are making it into food webs, and they seem to be important to these fish, although we don't have really much information as to what level of importance those nutrients might be. So all this stimulatory effect I'm talking about, all this bottom-up processes had to be good for sockeye, right? Well, when we do our juvenile hydroacoustic control surveys, we found that the lowest abundances of fish, the lowest sizes of fish were on record were found in that particular year. And you can see here that there was anywhere from a 27 to a 45% in those two dominant and subdominant cycle lines when they were rearing in the lake in terms of their weight loss, and 7 to 16% reduction in length. These are substantial size reductions which may confer survival disadvantages. Looking at freshwater survival, this is measured as the amount of fall fry, because we do our hydroacoustic enumerations both in summer and fall, but looking over that growing period window from emergence to fall fry residence, we can see that the survival to the fall fry stage was amongst the lowest on record also in Quesnel Lake. So these fish were not doing particularly well during the dominant and subdominant year classes in the system at those peak escapements. So let's move through the trophic levels in the different parts of the food web to understand if there's anything from a bottom-up focus that can inform us on this. And so sockeye salmon tend to be near obligate planktivores, meaning they focus in on zooplankton in their nursery lakes. A variety of, they tend to target a variety of organisms, but there tends to be a large focus on a particular organism Daphnia. There's a variety of species within that genus, but Daphnia tends to be a really efficient transfer of energy up through the food web to juvenile sockeye. And what we see in the big year compared to the before and after periods was a substantial decrease in abundance in biomass at the end of the fall period. So on the left-hand panel here, we see total zooplankton biomass in the water column, and on the right-hand side, we see that Daphnia, that really good and efficient grazer that sockeye really tend to key in on the right. Both were hammered. Both were drawn down to very low abundance relative to either historical or periods since, which you would predict based on a lot of hungry mouths within the system. You had a lot of top-down forcing from strong foraging, and so while that didn't necessarily confer to biomass, as you saw in the previous slide, they were really queuing in on zooplankton. Zooplankton were impacted, which in such a large lake is quite astounding to see. But we do see it across the major producers in the Fraser. Looking at size relationships, juvenile sockeye tend to be size selective in their prey selection, choosing the biggest, the fattest that they can get their mouths around. And so if you look at the major taxa, there were size reductions in all groups across during the peak escapement year there, suggesting that there was a strong cropping and size selective cropping of zooplankton resources within the lake. So we saw that there was huge exploitation of the intermediate trophic level, the zooplankton, and but the fish didn't really get all that big. So is it a simple density dependent relationship, or are there other processes that may be going on within the lake that could influence this? So let's go another rung down in the energy flow into phytoplankton, the autotrophic producers, the ones that harness those nutrients and pump them up into zooplankton production, ultimately sockeye. If we look at the functional size classes of algae, which is based on simply cell either volume or in this case size, we can see that there was disproportionate response during those peak escapements in the bacterial community, which is not necessarily unexpected. Usually when trophic status shifts toward more nutrient-rich conditions, you have a bacterial response. But what we really saw was an exorbitant response in the microplankton. And this can be iffy for sockeye because if you're thinking about a zooplankton floating around in a lake and its ability to actually eat phytoplankton within the system, size matters. And so microplankton are the bigger, the greater than 20 micron fraction, which, especially when they're chain forming, zooplankton have a hard time getting their mouth around. And so we had this massive response in microplankton, not necessarily a bad thing, but I'll tell you in this case it was. It was largely driven by diatoms. Again, those are those algae that incorporate silica or glass into their cell walls, which I don't mean to give the impression that they're bad fish food or bad zooplankton food, but in certain instances where they're big and the silicification or the amount of silica they incorporate is large, they can cause problems for energy flows. And in this case, the response was a particular taxon, which hasn't really been considered as a problem largely for grazers in the past, but it does have a number of characteristics which may have made it problematic for energy transfers within Quesnel Lake. And you can see amongst that diatom response, there was a massive response in this particular one, Tabellaria. The particular species that was in Quesnel at the time, I believe, is Tabellaria fenestrata. But nonetheless, it's a species that forms these long chains. Because it incorporates so much silica into its cell walls, it's very heavy, and it wants to sink out of suspension quite quickly. And so in this case, it forms long chains, and that allows wind-driven upwelling to keep it up in the photozone, keep it photosynthesizing. And it has a number of other features that may make it problematic. So if we look here relative to historical abundances of Tabellaria, which were negligible in the phytoplankton community in Quesnel Lake prior to the big escapement, we can see that Tabellaria dominated the plankton abundance throughout the entire growing season in that year that reflected the large subdominant run in Quesnel Lake. So what? Well, there may be reasons for this, and there may be reasons to believe that this was actually a problem for energy flows to sockeye, which may have contributed to those very small sizes. And so if you think about ecological stoichiometry of nutrients, the signature of nutrients you're getting matters. Do you apply a universal fertilizer for your grass needs, for your tropical plants, for your flowers, all that? No, they need different sources or different proportions of nitrogen and phosphorus for vegetative growth, for flowering growth, etc. The same applies to algae. And so in this case, Pacific salmon tend to have a nitrogen to phosphorus ratio of around 12 to 1 to 15 to 1, depending on the species and the area. Well, algal growth is limited either by nitrogen or phosphorus, depending on where you are on that continuum. And so if you're less than 20 to 1, you tend to be more limited by nitrogen. And if you're greater than 50 to 1, you tend to be limited by phosphorus. And in most cases, around the world, freshwaters are at least chronically limited by phosphorus. The story, of course, is more complex, but in the general sense here, we're receiving a source of nutrients, and in the case of a massive escapement like happened in 2001, 2002 in Quesnel, one that could have shifted the balance of nutrient sources for algae in favor of salmon-derived nutrients or marine-derived nutrients, it may have favored tabellaria, and I'll tell you why. You know, when you look at large calibration sets, this one's from the US EPA, tabellaria tends to be found in waters where you have slight to extreme nitrogen limitation. So there's reason to believe that the actual source of the nutrients from fish may have stimulated this particular alga when nutrient loading from salmon became high. It's a low N to P specialist, as I mentioned. It has the potential to be grazing resistant by zooplankton in lakes and a reduced food quality because of the colony formation here. You can see each one of these is an individual cell. Each organism is unicellular, but it forms these long chains which zooplankton have a hard time getting their mouth around. It also, as I mentioned, is heavily silicified. So if you take your meal tonight and you stuff it in a wine bottle and try to get it in your face, you're going to have a hard time digesting those nutrients, right? Because the glass is going to interrupt the ability for you to digest those nutrients, and you have a hard time getting a wine bottle into your gullet. So this is just showing that effect. I'm being quite colloquial here, but tabellaria uniquely forms a large number of what we call girdle bands. And I'm trained diatomist, so I nerd out about these things, but nonetheless, you have all this silicification which makes the cell heavy, requires those chain-forming assemblages, and creates a large vacuous space of which photosynthetic material, which is the vegetative material that herbivores like zooplankton need to survive, it makes it a small component of the overall cell. So you've got a big cell, number one problem getting your mouth around, and the actual valuable material is a small component of the overall bio volume of the cell. And so that may contribute to a problem for energy flows to higher order organisms. Is there other evidence that this is an issue? Yeah, well, especially on the outer coast, when they were trying to fertilize lakes here in Canada, they kept, depending on the nitrogen to phosphorus ratio of the nutrients they were adding to the system, you had a strong stimulation of this guy here, and this is Rhizosolenia ariensis. It's a common diatom in coastal waters, but what it is it's extremely long, and you can see it has these large spines, which again is believed to be an anti-predation morphology adaptation. And so this is just showing some work from Kim Hyatt, who's a scientist in our science branch, showing that when at certain times of the year in certain lakes, they would stimulate Rhizosolenia, and it would represent a trophic dead end, a interruption of energy flow to juvenile sockeye as a result of the nutrient cocktail they were putting into those systems. It stands to reason that the nutrient cocktail coming from marine-derived nutrients could do the same thing. So the potential for a bottom-up interruption enforcing within Quesnel at high escapements. And if we look at what the fish were actually feeding on, we don't have a lot of information for diet for Quesnel, but if we look at what we do have, which is typically on subdominant or non-dominant runs here, you can see either indicated by an S or an N. Prior to those big years, and that's that one there, prior to those big years, juvenile sockeye were doing what juvenile sockeye do. They were keying in on cladoceran zooplankton, which tend to be high nutritional value, pound for pound. And that was largely, in the earlier years, Bosmina shifted towards Daphnia. As I mentioned, Daphnia is a really efficient source because it has high fatty acid contents and can deliver that nutrient. quite readily to sockeye. They call them the cows of the lake. They're these big floating, really efficient filter feeders which transfer energy quickly. And during that high escapement year, indicated here, you can see that the food web shifted at least towards the end of the growing period where this snapshot occurs towards Dicyclops. And Dicyclops is a totally different beast than any of the Podosera. It's evasive, it's motile, it darts around, and being visual feeders, sockeye have to chase these things down to eat them. So there's a huge bioenergetic consequence associated with tracking down Dicyclops as opposed to getting all these big sea cows or lake cows that are floating around in the environment. And you know, in terms of food quality, Dicyclops is not on par with Daphnia. So potentially a less efficient trophic energy delivery to sockeye salmon as a result of the change in the structural composition of the plankton community. Again, conferred, you know, these processes may have conferred a very small or a reduction in size for juvenile sockeye in the lake. We know that their survival wasn't great. We know that they were of small size. And there's been a wealth of studies that have shown that at least in certain systems, and this is showing across a number of systems, that the amount of size you accrue in freshwater before you go to the marine environment matters in terms of your overall survival. So this is an across system comparison. The R-squared isn't all that great, but it does show a positive relationship between size of smolts when they're going out to sea and their smolt to adult survival, so their success in the ocean environment. And well, like I mentioned, this doesn't hold true for all stocks, but for Quesnel it does. Size really seems to matter in this system, and you can see that if they accrue a larger fall fry rate, weight or length, we don't actually measure smolts in the system anymore, but if the sort of returns per 1,000 fry as an estimate of post-fall fry survival is somewhat strongly correlated with the size they accrue in freshwater up until that period. It makes sense because they're feeding heavily during that growing period. They have to go through an overwinter period, which could be a sort of bottleneck for mortality or survival, and then of course the marine environment as well, which is a great filter. Getting close. And so if we look at that over time, we can see that the major runs actually didn't do all that well in the ocean as well, and it may be related to an interstage effect that started in freshwater. Of course, you know, 2009 they didn't do so hot either, and that was sort of the precipitation. The Cohen inquiry has been largely related to marine factors. So just to summarize the Quesnel story, you know, we've had massive escapement variability, especially in recent years. Peak escapements in 01 and 02 were followed by a decade or more of depressed abundance. In terms of the lake ecosystem itself, we saw very pronounced ecosystem structure and functional responses. Large-scale fertilization of Quesnel Lake for marine-derived nutrients was evident. Pronounced primary productivity responses, but unexpected energy flow relationships to higher trophic levels, and that may have been mediated by that particular alga tabularia. We know that trophic dead ends can really interrupt energy flows and have consequences for fish populations. May have been the case here. And there seem to be ultimate impacts on juvenile condition in the overwinter and marine survival phases. And one really unique finding is that this size really matters for Quesnel sockeye. If I did the same regressions for Shuswap or Chilco, that's not the case. So I just want to end the talk by talking a little bit about this concept of delayed density dependence, which of course is not novel by any stretch of the imagination, but this idea of cycle line interaction. Delayed density dependence is some sort of simple density dependence in the sort of dominant cycle line. It's a hypothetical mechanism for cyclic dominance in stocks, and it's a strong density-dependent relationship in freshwater, density-dependent relationship in the dominant cycle line, followed by some other factor that reinforces cyclic interactions. Disease on densely populated spawning grounds, increased buildup of predators in freshwater environments have been evoked, and potentially there's some evidence for severe interannual depletion of aquatic food webs. It's explicitly captured in Larkin stock recruit modeling, and that was a formative bit of information that came through in the Cohen inquiry to identify whether these processes are appearing in the Fraser. So I just want to point out a few things I know Carl will have issue with this particular slide, but nonetheless, the Larkin model did fit better than Ricker. So Ricker is a stock recruit model that incorporates density dependence, and the Larkin actually incorporates interactions between cycle lines. And I found it interesting that while Randall Peterman concluded that only Quesnel really showed strong evidence of delayed density dependence within Fraser's stocks, 9 out of the 19 stocks actually were fit better by a model that explicitly incorporated cycle line interaction. So it may occur in a number of stocks at lower levels of influence, but Quesnel may have been the most spectacular example of massive stock decline. This is some work of Jim Woody, who was the former chief scientist of the Pacific Salmon Commission, Mike LaPointe, who currently is, and Jeremy Hume, who was a former alumnus of our lab. What they were able to show, looking at either, again, circuli counts on the bottom, which is a measure of freshwater growth, or fall biomass in the fry, what you see is the dominant cycles show some sort of relationship, to effective female spawners in terms of biomass or circuli counts, right? You see that in the dominant line, and then subsequent lines show reduced growth. So here's the subdominant line in Quesnel Lake, and here's circuli counts from horsefly sockeye. You can see that those successive lines show reduced growth, and it's been related back to this cycle line interaction. We have never had this many fish before in terms of recorded history in the Fraser, but other people do, and so we can look to Alaska maybe for some information to try and inform what's happening at these large escapements in the Fraser. There's the large escapements in productivity in a report by Clark et al. In 2007 showed that there was productivity declines when you had back-to-back peak escapements. So escapements that exceeded the spawner capacity based on stock recruit for these systems. So you actually had 2 times S max, 2 times the maximum spawners back-to-back. And they detected delayed density dependence in 5 stocks with this quote-unquote over-escapement. You had recruits per spawner or falling below replacement, so a declining population associated with 2 to 5 years afterward, and in some extreme instances up to a decade. Sounds familiar? Fraser Lake, Alaska is a salmon system now, but it wasn't previously. It was prevented, migration was prevented by barrier falls. They put in a fish ladder, created a fish stock. It was documented in this particular paper in Canadian Journal of Fisheries and Aquatic Sciences back in the late 90s. But what they showed was they can build up a stock, but they can also collapse it. And so what happened here was you had a peak escapement in the 80s, which was followed by depressed abundance until the reset button was hit and the stock actually started to increase again. And they largely attributed this to some of the freshwater resources within the lake, overcropping the food web. And one of the major conclusions is that large escapements aren't necessarily independent, and they do influence each other, which is bearing true over a broader number of systems. They also, in the heyday of research, were able to do a lot of experiments where they'd add fish to systems. They'd monkey around with the nitrogen and phosphorus that they were adding to the system. So they were able to manipulate both top-down and bottom-up processes in these systems. And they were able to get some really persistent effects. So there was strong restructuring of zooplankton communities by sockeye. That's not necessarily groundbreaking, but one of the really interesting results of their study was that when you tried to reverse that with bottom-up stimulation, even at modest levels of planktivory or modest juvenile abundances within the lake, you couldn't turn the process around in a short order. In fact, the persistence on abundance of fish in those systems remained low through time, even though you were hitting them with lots of nutrients. So is this the mechanism at Quesnel, or for that matter, in broader sockeye nursery systems? People have looked at total zooplankton abundance in Quesnel, and this is just from the mid-80s, I guess, on through to 2007. You can see across a number of stations within Quesnel, and these ones represent the north and main arms of the lake. You can see that around 2001, 2002, there seemed to be actually a stimulatory effect on total Daphnia, at least in station 8, which is the north arm, a strong stimulation in overall zooplankton biomass. So that would say that zooplankton actually did better as a result of more fish, but the fish weren't doing very well. So that sort of supports that idea that there may have been a trophic disconnect. But it may not just be the total abundance that matters. When you're hungry now. And you need to have food to survive. And so this actually I produced this morning because I was rereading some stuff Carl sent me. And there's this pattern that seems to be bearing true across a number of the big systems in the Fraser during these peak escapements. It's shown in Shuswap, it's shown in Quesnel, and it's shown in Chilco. And what we see is during the dominant year, in black here, great zooplankton. I'm just showing Daphne here at a particular station for illustration. You can see strong increases in biomass throughout the growing season, likely conferring a strong energy contribution to juvenile sockeye getting big and fat, right? Unless there's some sort of mechanism that interrupts that, like the energy flow disconnect. In the subdominant line, look at the zooplankton biomass over that same time window. You can see that it's about half of what it was in the dominant cycle line. Those cycle lines tend to come back at very much reduced abundance compared to dominant lines. And then what happens in the first non-dominant line is you see that this is the pattern that seems to be reproducing across big systems with large escapements, is you get this retardation or this slowing down of the rise of zooplankton. This is exactly when fish are starting to move offshore. So juvenile sockeye are emerging, they're buttoning up, they're actually starting to forage, they're no longer relying on their internal yolk sacs, and they're starting to forage, right? And they need to move into the pelagic environment. And it's likely that the phenology of the food availability matters. And what we're seeing across all these systems is this delay in that subsequent non-dominant line of food availability as these fish move offshore. And if you remember back to what happened at Quesnel, during these exact years, we had the lowest freshwater survival to fall fry on record. And they may not actually be able to have the food web resources that are available to them at the time that they need them. This really shows strongly across Shuswap Lake and again in Chilco Lake with recent increases in escapement. So that's it's an incomplete story, but this is now where we're going to try and understand the relative influences of top-down and bottom-up, and there may be something to this phenology argument, which may be changing for a number of reasons, including climate change. So just to summarize the talk, I'm sorry I'm a little bit over time, that's my modus operandi is to ramble on. But Fraser River escapements have been increasing substantially beyond our window of understanding from a fisheries management context and also from a scientific context. And there's evidence from some systems that overescapement may be based on simple habitat, or overescapement may be exceeding habitat capacity. We show these strong trophic interactions in Quesnel. I won't go through all of them here, but just to say that there was strong bottom-up stimulation, potential trophic disconnects, and huge top-down forcings, which may have impacted freshwater and subsequent marine survival. And I think we need to start thinking a little bit more critically about large escapements and what that may mean for stock persistence. Again, while the co-inquiry suggested that only Quesnel showed striking evidence, 9 out of 19 systems were fit better by a model that incorporated delayed density dependence or cycle line interaction. So are there concerns for other stocks? There are large experiments unfolding before our eyes, and I think we're going to learn a lot about how stock dynamics in the Fraser occur over the next few years. Thanks.
Sorry about the mic. I hope that wasn't too disturbing. We have time for one question, but I think Dan will be around the afternoon a bit, so just approach him and see if you can talk to him after. Go, Carl.
No, just a kind of a question and comment. The Peterman-Dorner analysis, there's a mistake in it. I haven't tracked it down, but the strongest delayed density effects are actually in Adams, not at Quesnel. I don't know what the hell mistake Bridget made, but it's they did something wrong there.
Yeah, you mentioned that before.
Yeah. The other thing is the problem with the zooplankton interaction hypothesis for delayed density dependence is it doesn't explain the really poor performance of the second off-cycle line.
Right.
In Adams, at least. The second off-cycle did start to recover in Quesnel as the cycle broke down, but not in Adams. Your last slide showed this with the high zooplankton Daphne abundance in the fall of the first off-cycle one. So that means a good carryover to the next spring in zooplankton of the right kind. So the second off-cycle shouldn't perform poorly under that food hypothesis.
Sorry, I should have plotted that.
I think you should. It should come up about like the dominant line, or even faster, if I remember Jeremy stated.
Are you and I meeting after this?
Yeah.
Good. Let's look at that then for sure. But we are seeing this retardation effect, and I don't think it's inconsequential as far
The
as.
second off. Well, the question is it showing up on the second off-cycle line? Because my recollection from looking at Jeremy's data was no.
Let's check it out. Yeah, I couldn't tell you offhand right now. As I say, I just made this morning to confirm that. But okay, so cyclic dominance occurs across a diversity of freshwater ecosystems. The Adams is probably the most spectacular example of that. The potential for bottom-up processes to affect, forcing of stock abundance, I think is largely related to the resilience of those ecosystems. It may not be the overall factor, right? And as you mentioned earlier, there's been substantial debate between bottom-up, top-down, and other things, true top-down, like build up of predator populations. But all that to say, a lake is not a lake is not a lake, and I think some of the trends that we're trying to draw across ecosystems and stocks, like Shuswap versus Quesnel, are just not valid. Shuswap is a lake that's incredibly fertilized by things other than marine-derived nutrients. It has this sort of constant bottom-up forcing from agricultural inputs, from septics, from all kinds of sources. We just went through this with the Shuswap integrated planning process. It became very clear that there's other factors that are producing fish there, right? And of course, there's other mechanisms that may influence overall survival in freshwater. Chilco is another example, and that's one where I thought, holy jumpins, we had all these fish return in 2010, and that lake is not prepared from a trophic perspective to produce that many fish, I didn't think, right? So you look at it and you say, okay, well, but what's the limiting factor? Is it rearing in Chilco? And it has to be to a certain extent, but these fish all spawn at the outlet. The principal spawning environment is at the outlet in Chilco. You're not getting the marine-derived nutrient subsidies, and you're getting a lot of red disturbance or red uptake. I've stood there and watched it, right? One sockeye will move in and put down a red, and the other one will come right over it a day later and just tear it up and make their own. So I think there's a number of different forcings that are going on. We can't necessarily draw generalities across stocks, but totally looking forward to having a broader discussion on that.
Thank you very much.